Welcome everyone. Today we are diving into chapter 4, which covers the conquest of infectious diseases. We will explore the history of these diseases, the biology behind them, and the public health measures used to control them. Let's get started. Before we begin, take a look at the key terms on the screen. Throughout this lecture in the part 1 and part 2, we will be discussed concepts like aerosols, carrier state, bacteria, chain of infections, contact tracing, herd immunity, immunization, methods of transmission, parasites, pathogen, quarantine, reservoir, susceptible host, vector, vaccination, and viruses. Keep these definitions in your mind as we moved forward. The historical toll of infectious diseases. Throughout human history, up until the early 20th century, infectious diseases were the leading cause of human death. To give you an idea of the sheer scale of this threat, the Bubonic Plague, known as the Black Death, is said to have wiped out as much as 75% of the population in Europe and Asia during the 14th century. Later, in the mid of 19th century, tuberculosis became the number one killer in England. In the United States, in 19th century, in New York City, background mortality rates were largely driven by tuberculosis, by typhoid, and various respiratory and gastrointestinal diseases. We can actually visualize this historical impact. Here in this figure, showing the death rates for the population of New York City over a vast period from 1804, like here, 1805 to 2017. 2017. As you can see, the early years on the chart are marked by massive, terrifying spikes. These peaks represent brutal epidemic of smallpox, yellow fever, and influenza. Notice how cholera epidemics swept through the city every few years, killing many people in each wave. But look at the dramatic decline as we move into the 20th century. There's a dramatic decline of the peak. How did we achieve this incredible drop? Let's look at the conquest of infectious diseases. These terrifying diseases were largely conquered not just by doctors, but through fundamental public health measures. This included the purification of water, the proper disposal of sewage, pasteurization of milk, and the rollout of immunization program. Improved nutrition and better personal hygiene were also critical factors. Later, the discovery and introduction of an antibiotic in 1940s played a major role in dropping those mortality rates even further. The incredible success actually brings us to an interesting paradox, which we will discuss next. The era of complacency. Because these public health measures were so successful by the 1960s, the threat of infectious disease seemed to have been reduced to a minor nuisance. The intense fear, drama, and excitement that drove early public health efforts vanished, and the field started seem a routine and boring. In fact, according to the Institute of Medicine, during the 1960s and 1970s, public health began falling into disarray simply because of complacency. Now, to truly understand how we fight these diseases, we must understand what causes them. Let's together explore the agents of infections. Infectious agents. Major diseases are primarily caused by three types of agents. Number one, bacteria. Number two, viruses. Number three, parasites. The thrilling scientific realization that each of these diseases is caused by a specific microbe occurred during a period of great scientific excitement in the 1818s and 1890s. But think about the challenge. Humans are covered in billions of harmless bacteria on our skin, for example, in our throat, and in our intestine. So, how did scientists prove which specific bacteria cause a disease? Koch's postulates. German physician named Robert Koch developed a strict set of rules to prove that specific organisms caused specific disease. These are known as Koch's postulates. First rule, the organisms must be present in every single case of the disease. The second rule, the organisms must be isolated and grown in the laboratory. Third rule, when you inject that laboratory grown culture into susceptible test animal, they must develop the disease. And the rule number four, the organisms or the organism must be isolated again from those newly infected animal. Cook, the scientist Cook applied these exact rules in his proof that tubercle bacilli cause tuberculosis. Let's look at how we visually classify these bacteria. Classifying bacteria by shape. The scientist Koch also developed techniques to classify bacteria by their shape and their propensity to be to be stained by various dyes. We have bacilli, which are bacteria that appear rod shaped under the microscope. Bacilli are responsible for disease like tuberculosis, like cholera, like plague, like typhoid, like tetanus, and diphtheria also. Then we have cocci, which are round shaped bacteria. These cause conditions like strep throat, scarlet fever, wound infection, and pneumonia. Finally, there are spirochetes, which are cork corkscrew shaped, like here, such as the bacterium that cause syphilis. But what about diseases where no bacteria could be found? For some infectious disease like smallpox, scientists could not find a bacterial agent. Early attempts to isolate the microorganisms failed because the agent causing the disease could pass through the finest available filters and could not be observed in any existing microscope. These mysterious pathogens became known as filterable agent or viruses. We finally understood their true nature in 1935 when the American scientist Stanley successfully crystallized the tobacco mosaic virus. So, what exactly is a virus? Let's find out. The characteristic of viruses. Unlike bacteria, which are living cell, living single-celled organisms, viruses are not complete cells. They are complexes of nucleic acid and protein that completely lack of machinery to reproduce themselves. To reproduce, they have to take control of a host cell's machinery, which often kills the host cell in the process. They are remarkably resilient. They can survive extreme conditions like treatment with alcohol and drying in a vacuum and become active again when injected into a living cell. Human diseases caused by viruses include smallpox, yellow fever, polio, rabies, AIDS, and the common cold. Now, let's briefly touch on the third category, the parasites. Parasites are the final major category. These include protozoa, which are single celled animal that can live as parasites in the human body. They cause diseases like malaria, which is spread by mosquitoes, as well as cryptosporidosis and gardiasis. Then we have various worms, roundworm, tapeworms, hookworms, and pinworms, which are actually the most common source of human infections in the world. However, except for pinworms, they are not common causes of disease today. Next, we need to understand how these agents moved from person to person. Infectious diseases spread through several distinct routes. First, through aerosols. Respiratory infections like colds, influenza, and tuberculosis are transmitted through the air on water droplets produced when an infected person coughs or sneezes. They can also spread indirectly via objects. The infected person touch a doorknob or towel and the pathogen is picked up by the next person who, who touches the object and transfer it to their nose. Tragically, early European settlers made use of this rule as a primitive form of biological warfare, giving Native Americans blankets used by smallpox patients, which decimated populations that had no immunity. Let's look at the other routes. Gastrointestinal infections like cholera and cryptosporidosis generally spread via fecal oral routes. This happens when fecal matter from an infected person reaches the mouth of an uninfected person, usually due to poor personal hygiene or the contamination of drinking water from inadequate sanitary system. Then we have vector-borne disease like malaria, yellow fever, and West Nile encephalitis. These generally use a more complex route from person to person, most often traveling through an insect vector. But the timing of transmission is also crucial, as we will see next. So the time during which a patient is capable of transmitting an infection varies widely from one disease to another. Some diseases like tuberculosis or common cold are most likely to be transmitted during the most symptomatic phase, when the patient is actively coughing and sneezing. Others, such as measles and mumps, are most communicable during the day or two before noticeable symptoms even developed. And incredibly, a few diseases can exist in carrier state, in which infected person can transmit the disease without ever having symptoms. The most infamous example of this is Typhoid Mary. Let's look at her story. Mary Mallon worked as a cook in wealthy New York households in the early 19th century. Because of her occupation, she spread typhoid fever via fecal oral route to growing number of family members, some fatally. Because she was perfectly healthy, she firstly resisted medical tested and refused to accept the result that proved she was carrier. Eventually, she had to be in, incarcerated by a New York Health Department for the rest of her life to stop her from taking jobs as a cook. The discovery of antibiotics later made it possible to eliminate the bacteria in typhoid carriers, but it came too late to help Mary. Now that we understand the agents and the transmission, let's look at the big picture, the chain of infections. The chain of infection, the core of the public health approach to controlling these infectious diseases is to interrupt what we call the chain of infections. This chain, as you see in this figure, the, map the pattern by which a disease moved from person to person. It composed for several links: the pathogens, the reservoir, the place of exit, the method of transmission, the port of entry, and the susceptible host. Our goals as public health professionals is to find whichever links are most vulnerable and break them. Let's break down those main links, starting with the first two: pathogen and reservoir. Link number one: as a pathogen, the specific viruses, bacterium, or parasites that causes the disease in humans. Link 2, reservoir. The reservoir, the place where the pathogen lives and multiplies. Some pathogens have a reservoir that is strictly human to human. Other infects non-human species. For example, rats. Rats are the reservoir for the plague, and bats and raccoons are reservoir for rabies. Contaminated water or food may also serve as a reservoir of some human diseases. Now we look at how the pathogen gets to its target. Link 3 is the method of transmission. The pathogens need needs a way to travel to new host, whatever it's through the air, contaminated food, drinking water, sexual contacts, or a vector like aphilia. Finally, link number 4 is the susceptible host. Even the pathogens gains entry, that new host must be susceptible. The immunity might develop as a result of previous exposure, or the host may naturally lack susceptibility. For example, the canine distemper virus simply does not infect humans. So, how do we use this chain to fight disease? Let's look at our interventions. We attack the vulnerable links. So, the interpreting the chain links 1 and 2. At link 1, we can kill the pathogen directly, for example, by using an antibiotic to destroy disease causing bacteria. At link 2, reservoir, we can eliminate the reservoir that harbors the pathogens. If rats harbor the plague, we control rat populations in cities by picking up garbage. We ensure adequate water and sewage treatment to eliminate the reservoirs of waterborne diseases. What about link 3 and link 4? At link 3, we prevent transmission from the one host to the another. This includes quarantining infected individuals, warning people to boil contaminated water, frequent hand washing, and to protect food and patients, and the use of condoms to block sexually transmitted pathogens in the non Muslim countries. At link 4, we increase the resistance of host primarily throughout the immunization. This is the all in this part 1. See you in the part 2 to complete chapter 4. Thank you and goodbye.